Hello, everyone. I'd like to also say thank you very much for inviting me to come and speak at this conference. My name's Claire Sadler. I'm a project toxicologist. I have about 15 years' experience in the pharmaceutical industry. I've worked across a number of different indications, in oncology, inflammation, and infection. And I have some, I used to work at AstraZeneca for about 10 years, and I can see some ex-colleagues of mine in the audience. Hopefully they won't heckle me too much. I currently work at a company called Iconics, and we are actually toxicology consultants. And just a very interesting talk from Terry, and I will touch on some of those points going forward. So in my experience working in projects for many years, sometimes the non-clinical safety is viewed as a hurdle, something that a project has to get over or negotiate towards the end of a program just to get through it. Or some may consider it just to be a box to be ticked. There are guidelines out there, there are suggested designs on studies, and you just have to fill out all those boxes, and then you're free to go. And even, in my experience, sometimes we're seen as a bit of a grim reaper. We can kill your projects dead if we have toxicity issues which can't be mitigated or negotiated past. But really what we're here to do is try and help move the project forward. We're all kind of trying to do the same thing. We're trying to get antibacterials into patients and onto the market to help combat the issues that we're currently facing. And what we're trying to do really is get medicines to patients as quickly and as safely as possible. Just touching on the guidelines that Terry actually mentioned there, if we look at what they actually say we're trying to do in this non-clinical evaluation time, we're just trying to characterize the toxic effects. We're trying to identify the target organs of toxicity. We're trying to understand any dose dependence and the relationship to exposure. Is that a steep dose response curve, or do we have a bit of wiggle room there? We're trying to also understand the potential reversibility of any toxicities. And from that information, we use it to set the safe starting dose. We determine the dose range that's going to be used in clinical trials, so determine the exposure caps that you may be subjected to. And we're trying to identify the parameters for clinical monitoring. Today I'm focusing on small molecules, but I've also put the front page of the ICHS 6 guidelines, which is for biologicals. So there is different guidance for biologicals as there are for small molecules. A lot of the principles for toxicology are the same, but just to be aware that some things are a bit different. So Terry really has been talking about the regulatory side of toxicology, and from this diagram here, which is you'll be all familiar with of the drug development process, toxicology is actually one of the few functions which is working throughout that development chain. The red circle there is really highlighting the regulatory toxicology part of that chain. So looking at a compound that's been selected for development, going through GLP toxicity testing, and going through the clinical trials. And through those clinical trials of both phase 1, phase 2, and phase 3, there are different regulatory required non-clinical studies that have to be conducted to support those different phases as they progress. And although there are guidelines that give you an indication of what those studies should look like, it's really important to design those programs to meet your program's requirements. You need to make sure you consider things like the duration of dosing. Is this an acute treatment or chronic? How are you going to administer this drug? Particularly for antibacterials, the combination therapies, and when are you going to combine with other agents, and how much is known about those? Which demographic or population are you going to be going in, and when? Unfortunately, infections can hit anybody, and women of childbearing potential are an important demographic that needs to be covered. And also children, so we often have to treat infants and neonates. Again, when do you need to have the tox package to support going into that population? But today what I'm actually going to focus on is the discovery end of the pipeline. So in discovery, we have a slightly different aim maybe than from regulatory, where you've got one compound to work through and you're just trying to understand the risks. In discovery, what we're trying to do is identify any potential hazards early, try and design experimental plans to assess those, and really try to influence candidate selection so that we choose the best molecule possible to give you the best chance of success. In discovery toxicology, we work straight from the target identification all the way up to candidate selection. And as mentioned in the previous talks today, this is all an integrated process. So we have to integrate with the chemistry, the physical chemistry properties that were covered this morning, think about obviously efficacy and the spectrum and the microbiology, the DMPK properties, if you can't get it in, it's not going to work, and also the toxicology. And often if you change one element of these, it will change some of the others. So it's really about trying to find that sweet spot or the balance between all those different elements to get the best drug possible. So if we think about discovery toxicology, sometimes it's useful to break it into 3 areas. We have the target and toxicity liabilities associated with that. We have chemistry, and then we have patient. These are all interlinked, but you can look at them all slightly separately. So for target, aim of target assessment really is to understand the normal physiology of a target. If you're going to modulate it for a disease, what happens when you modulate it in the rest of the body? Is there some adverse event that will occur? And we can use data, in silico data, literature reviews to try and understand that normal physiology, look at things like orthologs, expression analysis, pathway mapping, the phenotypes, knockout animals and human mutations, although it doesn't always occur as they are, it gives you an indication of maybe where to look. And also anything you know about competitor compounds, maybe somebody's looked at something before. There's also in vitro tools we can look at enzyme selectivity assays, general cytotoxicity assays are useful, and also organ-specific assays. And then as the program progresses, you may have some in vivo data. You can take information from PK data, PK studies, efficacy studies, or investigational tox studies. You just need to know what question you're trying to ask. Is there something you know about this target that makes you particularly concerned about a particular organ or a particular endpoint? And can you incorporate that into some of your early in vivo studies? Now, you may think, being antibacterial, that actually the primary target isn't so important. Often you're trying to aim to hit something which is bacterial specific or bacterial selective, and therefore a human or a mammalian target isn't really relevant. And that might be the case depending on which target you're going for, but obviously this is just a slide of normal antibiotic modes of actions or common antibiotic modes of action. And if you're going for something on the cell wall, yeah, you're probably right. You're probably not going to be so concerned about your primary target. But if you were going for something like a nucleic acid synthesis target, topoisomerases, then actually you may find that mammalian cross-reactivity is something of concern or something that you need to be aware of. And as more and more novel targets are being looked at, more consideration has to be made for this. Now, I do appreciate that, as we heard this morning actually, the movement towards more phenotypic screens is around, and therefore you're not necessarily going to know what your target is, your molecular target from the beginning. But that's something that hopefully will be built as the program progresses. This is just some examples really of, so quinolones are known to inhibit bacterial type 2 topoisomerases, but they can also have activity against mammalian topoisomerases. And the top paper there is just showing how oxafloxacin causes rat testicular toxicity. It inhibits the DNA during the meiosis in the testes. And so although it's preferentially hitting bacterial type 2 topoisomerases, it does have impact in the mammalian system. The bottom paper there is really talking about 4 quinolones, which inhibit mitochondrial DNA, because mitochondrial DNA has some similarities to bacterial DNA, and so sometimes can have a bit of an issue when you're hitting that target. In terms of a target safety assessment cascade, in my experience, it's really about trying to identify potential hazards by inhibiting any orthologs. Again, looking at that literature to try and pull that out. And then developing in vitro assays to try and understand that selectivity window that you might have. One of the difficulties in this area really is when you're looking at activity in whole cell systems, so you're looking at bacterial activity versus activity in mammalian cell, is how do you compare MICs versus IC50s? These assays, in vitro assays, are very different. The formats are different. The endpoints are different. The culture systems are different. So can you really make a good judgment of the risk from that assay? So how big a window do you need when you're looking for fold selectivity? Are you looking for 10-fold? Are you looking for 100-fold? Are you looking for 1,000-fold? And really, it's not necessarily possible to identify that from in vitro work. You really need to do further work to identify the risks, potentially doing in vivo work to understand, okay, I've got a 100-fold selectivity window. I know what target toxicity I might expect to see if I was hitting that mammalian target, what actually happens when it goes into a whole animal system. The second area to think about is your chemistry, and this is definitely a consideration for anti-infectives. So you're really trying to understand your off-target liabilities and any potential baggage that you're carrying, which may limit your ability to explore your efficacious doses in the clinic. So again, using in silico, SAR databases, historical knowledge, and of course, as the talk this morning mentioned, thinking about AI, using in vitro safety screens, and I'll talk a little bit more about potential ones to use in the next slide. And again, using in vivo studies, any in vivo studies you're doing, thinking about what liabilities you may be carrying, and is there any way you can assess them early. And here, the real aim is try and identify those potential hazards early, so that you've still got time to design them out with the chemistry. So an example here is of some of the major areas for antibacterials where you may want to consider doing in vitro screening. So cardiac ion channels, a lot of historical knowledge about this chemical class and its potential to inhibit cardiac ion channels. HERG is obviously commonly known. But I'd also urge you not to just think about HERG. You need to think about the other ion channels involved in the system, things like sodium and calcium channels. You have secondary pharmacology screens. Again, just looking at general promiscuity early in a program can really help you understand the potential risks you may be carrying. And interesting, a question that was this morning about physical chemical properties in vitro screening, toxicity screening. So in my experience, if you have a compound that has high lipophilicity, then you have a very promiscuous secondary pharm profile. So that's one of the examples where the physical chem properties of the compound can really influence that off-target profile. For antibacterials, genetic toxicity is very important. We have to think about the mutagenicity, and as mentioned this morning, there's a lot of structural alerts that we understand how that links into direct damage of DNA. But also with antibacterials, there's some primary targets where they're linked with the way they interact with DNA and causing signals in the genotox assays. So again, something to understand early on. Mitochondrial toxicity, as I mentioned before, is a common toxicity with antibacterials, and again, it can be an issue. And in my experience, using a general cytotoxicity in mammalian cell, just to understand how potent your compound is versus bacterial, can be quite useful to understand the potential hazards you may encounter. And something just to note, again, top dose to be used. So most indications would use 10 micromolar in an in vitro screen as their top dose concentration. It will be well excess of where they plan to be in the clinic. In my experience in antibacterials, 10 micromolar free is not necessarily in well excess of where you may need to be for in the clinic. So when you're looking at these in vitro screens, you need to think about what that top dose concentration is, and then you need to think about the solubility of your compound at that higher doses. So again, that's where physico-chem properties can really influence how you use these assays. So again, the in vitro assays here are really screens to try and identify potential hazards early, but all of them, you can get an IC50, but it doesn't tell you the whole story. You have to follow those leads and try and understand what the risks are. And I've just got a couple of examples really of programs that I've been involved with. So as we know, MBTIs, pharmacophores are well associated with cardiovascular liabilities. And this is from the AstraZeneca group over in Boston, where looking at novel aminopyrimidines and their inhibition of topoisomerase and their liability in the cardiac channel. So on the top left, you have a compound 7, which was the HERG IC50 was 44 micromolar, which in other indications you would be happy as Larry, that's really high, but again in infection is not necessarily high enough. And bear in mind for HERG as well, IC50 isn't really the relevant number, although that's the one that everyone reports. And IC10 is when you start seeing activity. They assessed this compound in vivo in a guinea pig map study to look for QT prolongation. And you could see QT prolongation was demonstrated at less than 20 micromolar free. The effective therapeutic plasma concentration that was being calculated was around 2 to 10 micromolar free. So you can see your window there between an effect and where you need to be is pretty small. And so it was considered that this would have a high risk for QT prolongation in man. So there was a lot of effort put in to try and reduce that HERG inhibition, and that was actually done by reducing the pKa, so again, how physical chemical properties link in, whilst trying to maintain the other properties of the compound. And in the bottom right corner, they had a HERG IC50 of 233 micromolar. They assessed it again in a guinea pig map assay, and at the highest dose they tested, it had no QT prolongation. This second program is from the AstraZeneca group over in Bangalore when we were looking for an antimalarial candidate. The top left graph shows you an early compound where it was shown to have very promiscuous activity against secondary farm profile. The lines on this curve, I've just annotated some of the points. The far left point that you can see is acetylcholinesterase inhibition, and as we go through the different hits that it had activity also at muscarinic receptors and adrenoreceptors. And the solid line on that graph, I don't know if it shows very well on the screen, there's a solid line on the right-hand side, which is where they predicted they needed to be for efficacy. So basically, we were getting very high coverage of these off-targets within our efficacious range. This was borne out by going into an in vivo study and seeing if we saw any toxicity, which we did, indicating that the selectivity profile of off-targets wasn't good enough. So a lot of work was done to try and move that selectivity profile. And on the bottom graph, you can see they actually moved the acetylcholinesterase, which was the focus of the SAR, over to the right-hand side. So they took away that activity. And interestingly as well, we also increased the potency. So the solid line has moved far to the left. So as well as getting rid of one of the off-targets, they increased the potency, so they increased that window of safety. It doesn't really matter how that window is brought in, whether it's you get rid of off-targets or you increase your potency. It's just about expanding that window as good as possible. This also went into in vivo testing, where we found that we got good tolerability at doses that was required. And this third example was from the team over in AstraZeneca Bangalore as well, where here we were looking at nitroarenes and for tuberculosis. And nitroarenes are known to carry the risk of mutagenicity. Again, it comes down to their physical chem properties and their nitrogens. And they had really good promising activity, but this was obviously a concern if they were also mutagenic. So we investigated cutting down the AIMS assay, basically, to be able to look at a number of compounds early on in the program. For those of you that aren't aware, an AIMS assay, which is the regulatory-required mutagenicity assay, requires up to 400 mg of compound to assess. So although it's in vitro, it's compound hungry. 400 mg of compound early in a program is a lot of compound. So we adapted the assay to be able to use only 10 mg of compound, which made it a lot more tractable, to be able to look at matched pairs and work through the SAR to reduce the mutagenicity. So coming on to the third part of when we think about the toxicology, which is the patient. So we can identify the hazards early, we can look at the target-related hazards, we can look at the chemistry-related hazards, but really we need to put those hazards into context with the patient population, and we need to think about the risk versus the benefit for that population. So in order to do that, it's really essential to understand where you're going with the program. What's the clinical plan? Are we going into healthy volunteers or are we going straight into patients? With a lot of anti-infectives, we have to go into healthy volunteers to get the PK initially. The downside of that is healthy volunteers can't tolerate a toxicity that maybe a patient who is in a life-threatening condition would tolerate. We have to think about the route of administration. Are we going oral? Are we going IV? If we're going IV and we have a toxicity which we can monitor and we take away the drug very quickly, does that also mean the toxicity disappears? If we're going orally, that might not be as easy to do. Are we treating the patients in the hospital, or are we treating them in the community? If they're hospitalised, they'll be highly monitored. We can then address any issues as they arise. If they're in the community, they have a lot less monitoring, and we have to think about the risks associated. We have to think about the duration of treatment. Are we treating acutely? Is it 7 days treatment for a serious hospital infection? Or is it a chronic treatment? Is it tuberculosis where we'll be treating them for 6 to 18 months? A toxicity which is maybe tolerable and have a good safety window for 7 days, because we can show things like reversibility, may be looked at very differently if it's something we're treating for 18 months. We also have to think about the age of the patient. Are they elderly? What the comorbidities they'll also be having? Or are they young? If it's something that may affect fertility, say, an elderly patient may accept that tolerability risk, where a neonate, that may not be so acceptable. And all of this has to be put in with the calculated exposures required for efficacy. So any hazard we identify, it's really about where do we need to be for efficacy, what's the exposures required to understand that risk. And I appreciate early on in a program, it's a very difficult thing to answer, so we use surrogates, we look at MIC90s, we look at efficacy exposures, but the real assessment comes on when we can understand where we need to be in the clinic. This is a slide I quite like. It's an indication of how to look at hazard versus risk. So here we have 4 men in full protective gear looking at something in a field. I don't know what it is. They've assessed that. They've looked at that hazard. They've made a risk assessment, and they've decided they need to be fully kitted out to understand it. And then we've got somebody who's just stood there. He's exposing himself more than they, but he thinks that's okay. I don't know who's right. I don't know the circumstance, but it just shows the difference between looking at a hazard and understanding the risk of it. So one of the questions I'm often asked is, well, what do you do when? You're trying to develop a drug, you've got your efficacy to worry about, your microbiology activity, you've got your physical chem properties, you've got your DMPK properties, you've got your safety profile. It's a lot to try and shoehorn into a program where you have limited money, you have limited compound supplies, and you have limited time. And really, for me, it's about trying to understand early on, what do you think your potential hazards are? Build a bit of data, try and understand how real you think they are. And if you think they're real, you build on that some more, you add some more experiments in to understand the real risk assessment that you might have, and you try and mitigate around them. But it's an iterative process, and it's something that should be done right from the beginning all the way through to your candidate selection. Tox shouldn't be just done right at the end. So in summary, safety should be part of the whole drug design process. Identify hazards early, design experiments to understand them better, mitigate them if you can, put any risk you do find into context, and think about the risk versus benefit for your patients. And with all that, you hopefully will identify the best molecule to give you the best chance of success. Thank you very much.